everyone, welcome once again to the Revision Vault. Today we're going to be wrapping up the topic of gas exchange. This is part of the CIE syllabus for AS Level Biology. We're going to be covering all the criteria and making sure that we know everything that they could throw at us for this topic. So here I've written out, this is like directly from the syllabus, and I've written on the sides what I've covered in part 1 and part 2. So as you can see, part 2 is mainly filling in the gaps with a focus on the alveoli and the gas exchange. We're going to be going deeper into the gas exchange system. So in the first video, you would have seen that we focus primarily on the trachea, because that is the windpipe, as you may know it, or the initial entrance way for our air when we breathe in. Once this starts to enter our lungs, it is going to go from the trachea to the bronchi, to the bronchioles, to the alveoli. Then the gases will then diffuse into the capillary network or out of the capillary network, depending on which gas it is, and we'll be covering that in today's video. So, to start off with, we're going to be looking at the bronchus. Now, the bronchus has irregularly shaped cartilage plates. This is still hyaline cartilage, the same as what's in the trachea, and it is the distinctive feature of the bronchus. So, when we're looking at photomicrographs of the bronchus or diagrams, this is what we're going to be looking at to make sure that we can identify it as being the bronchus. And we can see these plates sort of surrounding the lumen here. It's in the blue. And if you look over here to the left, you can see a diagram of basically the entire gas exchange system. So the trachea is what we covered last time, and you can see these cartilage rings sort of surrounding it, and they are U-shaped, as you might remember. And here we have the primary bronchi and the secondary bronchi, tertiary bronchi. We don't need to worry about those terms. The main thing that we need to know is that there are two bronchi branching off of the trachea, and these then branch further into smaller and smaller and narrower tubes. Now, the bronchus is the singular version of bronchi, and it has pseudostratified ciliated columnar epithelium. Essentially, if you would have seen my last video, you would have seen that pseudostratified means that although it may look like the epithelial cells are staggered or in multiple layers, they are all in fact in one layer. And it's columnar epithelium, which means that it is the same cells as line the trachea. So at the moment, we have not seen any change in the cell lining. It has fewer mucus glands and goblet cells than in the trachea, and this is a pattern that you'll see throughout as we enter further and further and closer to the alveoli, because essentially we do not want infection to reach our alveoli. They are the most delicate parts of our gas exchange system, and if we do get pathogens coming into our alveoli, they could enter our bloodstream, which could lead to serious infection. And that is something that our gas exchange system has learnt to try and mitigate. So we have less and less mucus as we go further down the line, and therefore we have less mucus glands and goblet cells which secrete this mucus. You'll also notice, because cilia are the little hairs that we get on our epithelium that push the mucus up towards our trachea, towards our throat to then be swallowed. Therefore, we get less and less cilia further down as well. Now, the bronchus also contains connective tissue, which would be elastic fibers, smooth muscle, collagen, and blood vessels. So the elastic fibers and collagen are the connective tissue. The smooth muscle is present sort of around the lumen here as well, and the blood vessels, same as in the trachea, we do get them present throughout. They won't be shown in every photomicrograph because obviously these are cross sections and you may not receive a blood vessel in the image provided, but we need to be aware that they are present. Moving on to bronchioles. Now there are lots of different types of bronchioles, but primarily we need to know two. So the bronchioles come off from the bronchus, as you can see. As you can see here, the bronchioles come up from the bronchus, they break off, and from the bronchioles we then get to the alveoli. So the bronchioles get gradually smaller, and here we have it written out as the lobular bronchiole, terminal and respiratory, before we go on to the alveoli. Now, we only need to know these terms, the terminal bronchioles and the respiratory bronchioles. I know it may seem counterintuitive that terminal, which, indicates end, is actually not the end of the bronchioles, because after the terminal bronchioles, we have, in fact, the respiratory bronchioles, and from those we get onto the alveoli. So that may be something that will take some time for you to wrap your head around and get used to those terms being a little bit contradictory to what you'd imagine, but you'll get used to it eventually, don't worry. So the bronchioles have less cilia than in the trachea or the bronchi. As I've explained, it is because we want less and less mucus, and this is to prevent infection further down in the gas exchange system. They have no cartilage, and they have very few, or essentially none, goblet cells and mucus glands. Again, less mucus, less cilia, for the reasons stated previously. They have a thicker layer of smooth muscle than in the trachea or the bronchi, but they have thinner muscle strands, and in general their walls are thinner than when you look at the trachea or the bronchi, since they do contain cartilage, and that is quite a thick layer. And since we're lacking those in the bronchioles, and as I've said previously, they get smaller and smaller, the walls get thinner as well. And the terminal bronchioles have some smooth muscle, but respiratory bronchioles do not have smooth muscle, and they have less cilia than terminal bronchioles. So, like I've said, the wall is thinning as you go further closer to the alveoli. They may have a folded epithelium when its smooth muscle contracts, as you can see in this photomicrograph here. It's sort of these little folds within the inner layer facing the lumen, and that is because of the smooth muscle contracting, and that causes these folds to occur. If you imagine, if you took a piece of clothing and you were to scrunch it up and contract the outside, then the inside would be folding up. It's not the best example, but it's something that you can practically do. You could also do the same thing with a hair bobble, but it would have to be more the flexible one. And this does not happen in the respiratory bronchioles with no smooth muscle, because obviously smooth muscle is responsible for contraction and relaxation the same way we have muscles throughout our body, such as, like, the most common example is probably our bicep. As we contract, we lift our arm up. As we relax, our arm falls back down. And it's the same within our gas exchange system, and primarily in our bronchioles. They have ciliated columnar/cuboidal epithelium. So, because these are two different types of cells, you cannot have, well, you could, I suppose, have them alternating, but that's not the case. We have ciliated columnar in the terminal bronchioles and as you go further to the respiratory bronchioles, that's when the transition occurs, and you now have ciliated cuboidal epithelium. All the bronchioles contain elastic fibers and collagen, which is our connective tissue. We have smooth muscle on the terminal bronchioles and blood vessels, but we have no cartilage, which is different to the parts that we've seen previously. The bronchi and the trachea both contain cartilage, the bronchioles do not. And we won't be seeing the return of cartilage in the alveoli either. Moving on to alveoli. Now I've split this amongst 3 slides because we have quite a lot to cover here. As you may have heard in IGCSE or GCSE, the alveoli have many different adaptations that help make gas exchange and the diffusion of the gases more efficient. So the first adaptation that we generally learn is that they're 1 cell thick, and we know that these are squamous epithelial cells. So this essentially means that the cells are flattened and their nuclei do bulge slightly because of this. In case Cambridge asks you for the measurements, it is 0.1 to 0.5 micrometers thick, and it provides a shorter diffusion distance, which, as we know, increases the rate of diffusion. Because if you imagine you had to walk from one side of the room to another, it would take you less time than if you had to walk from one country to another country. Let's say if you were to walk from London to Edinburgh, that would be quite a distance compared to from one corner of your bedroom, which may then take you 6 meters if you have a big bedroom, to the next corner. Collectively, they provide a large surface area. Again, we know that this increases the rate of diffusion. Now, the walls have elastic fibers, which are made of the protein elastin. This is really important, and you may not have heard this in your previous years of study, so you need to be listening really carefully. The elastic fibers allow the alveoli to increase in volume without bursting. If you imagine a balloon and you were to blow it up with air as much as possible whilst you'd be increasing the surface area, it would eventually burst, most likely. And to prevent this, we don't want alveoli regularly bursting within our lungs. I imagine it would be painful, but more than that, it would have hemorrhaging and other severe consequences. So we want to prevent this, and we do this through the elastic fibers. If you imagine you were to wrap the balloon with an elastic band, it would be a very long one, but you would have this crisscross pattern that we can see in this image with the elastin fibers. Then if you were to blow the balloon up, as soon as you were to stop blowing, that air would come rushing out of that balloon. The reason being that there is elastic potential energy stored in those fibers. That's more going into physics, so you don't need to know the details of that. But you do need to know that the elastic fibers stretch during inhalation and recoil during exhalation. So the stretching increases the surface area, and the recoiling allows the expellation of air to be faster and more efficient. Okay, moving on to alveoli part 2. We have capillaries surrounding our alveoli, an entire capillary network in fact. And capillaries have a 5 to 10 micrometer diameter, and this is because the blood cells themselves have a diameter of around 7 micrometers. So this is just the right diameter to allow the blood cells to come through. And again, this decreases the diffusion distance, since the red blood cells are so close to the endothelium, which is the lining of the capillaries, and they are made of squamous epithelium the same way the alveoli are. As you can see, this is in this diagram, these cells surrounding the alveoli are the same as the cells surrounding the capillaries. It's just a special type called endothelium, but make sure you do not say that alveoli have endothelium, and you do not say that capillaries have squamous epithelium. You have to make that distinction in your exams. Now, as we can see, these gases very easily just go through this first one cell layer, this second cell layer, and right into the red blood cell, which is very, very close, basically touching this layer. And as you'll see in later photomicrographs, sometimes because the capillary lumen is so narrow, the red blood cells may have to curve even more, and that means they are definitely touching the capillary walls, and those gases can exit and enter as efficiently as possible. Now, the alveoli are surrounded by a network of capillaries, as I've said, and this maintains a steep diffusion gradient because they have a good blood supply, and this in turn increases the rate of diffusion. Now, this may be a challenging concept to understand, but if we imagine it like people needing to go on public transport and you're traveling on a train or a bus. Let's say a train, because I feel like trains are more regular. Now you're in the London Underground or wherever, and lots of people are traveling to the same place at the same time. If you have many trains coming, one after the other, those people will be able to get on the train faster and get to the place they need to be. Whereas if you only have a train once every 15 minutes, then the crowd will continue to expand, therefore meaning that once a train does come after 15 minutes, they will rush on as much as possible, but not all of the crowd will be able to get on. And that is the same concept with our gases. The more red blood cells that we have passing through the capillary, the easier it is for our carbon dioxide to unload and our oxygen to load. And our consistent bloodstream allows for this gas exchange to occur. However, the blood does flow slower in the capillaries, so even though we do want more trains for the people to be able to get on, we do not want them to be too fast. If you imagine a train coming and you only have 3 seconds to get on before it closes its doors and makes its way, even more people will not be able to get on that train, and it will not be as efficient as leaving the doors open for 15 seconds to allow it to fully pack and before it can leave. And it's just the same concept here. So the reason the blood slows down is because the combined or total cross-sectional area of the capillaries is largest than all of the blood vessels. For this, I'd have to draw it out for you to understand. If you can see, we have an artery coming out here and a vein coming out. The capillaries are sort of this entire network. So you break off into more and more capillaries from just a single artery, and that makes its way into a singular vein. So you have several capillaries in this network, in this section here. That means that the blood is rushing through at a constant speed here, but then it slows down because it has more entranceways and more tubes to get off. It's the same way as if you had a building and everyone was coming out of the same door. It'd all be rushing at a higher speed than if there were several doors for the crowd to get out of. Then they'd be able to go at a slower speed, but more people would be able to exit at that slow rate. Now, This allows for more time for the gases to diffuse and for equilibrium to be reached, and more complete gas exchange can occur. There's also good ventilation of the lungs and good circulation. That's another adaptation, and this maintains the necessary concentration gradients for oxygen and carbon dioxide, which are our main gases that are diffusing across these areas. And this, again, is concentration gradients. So the same way that good circulation provides a good concentration gradient, it is also like that with the ventilation. Again, I'll use the train metaphor or the train analogy. I think it's quite useful because if you'd imagine that the train was 50% full, then only 50% of people from that crowd could get onto that train to fill it up. Whereas if that train is 0% full, there are no people in that train. When it arrives at the station, many more people can get on that train. And it's the same with the ventilation, that we want to make sure that we get all of our carbon dioxide out of our bloodstream and all of our oxygen into our bloodstream. With higher concentration gradients, this diffusion will occur faster. Partial pressure. Now, this is maybe a new term for you. It certainly was for me when I was taking this course, because it's not something that you come across in year 11 biology. Essentially, partial pressure is the pressure that a single gas in a mixture would exert if it were the only gas present in the same space. Now, I know this may seem complicated, but I'll be honest, you don't really have to worry about it too much if you don't quite understand it. It's the pressure that the gas exerts on the container, or in this case, on the alveoli or the capillaries. And the partial pressure of a certain gas, it will not be affected by the partial pressures of the gases surrounding it. And the total pressure of a mixture is the sum of all the partial pressures of the individual gases. The pressure gradient is essentially the same as the concentration gradient. From a higher partial pressure, it goes to a lower partial pressure, and from a higher concentration, it goes to a lower concentration. It's the same sort of concept. And over here, you can see this is taken from our textbook. 13.9 kilopascals of oxygen is present in the alveoli, and that diffuses across to the 5.3 kilopascals that we have when the bloodstream just enters next to the alveoli. Here we have 6 kilopascals of carbon dioxide that moves into the alveoli, where it's just 5.3 kilopascals. And over here on the right, you can see that this has reached equilibrium because the partial pressures are the same in both the alveoli and the capillaries, or the red blood cells. I haven't come across any questions where it asks you to specify the partial pressures. I would memorize them just in case. That's what I did when taking the exams, but it never came up in my experience. Okay, alveoli part 3. This is the last part that we have to cover on alveoli. We have two types of cells in the alveoli. We have type 1 pneumocytes and type 2. Now, type 1 are the typical ones that you'd think of. So they're the squamous epithelium, they're thin with the bulging nuclei, and their function is solely gas exchange. They make up 95% of the alveolar surface area. Type 2 pneumocytes are cuboidal, and they make up 5% of the alveoli surface area. Their job is to secrete pulmonary surfactant. Now, we'll reach onto surfactant, we'll come to its function and what it is in a second. But first, we're going to cover surface tension. Surface tension you may have heard of as a common term. Because the alveoli are coated in liquid on their inner surface, the liquid molecules have cohesive forces which pull them together, and that is creating this surface tension which could cause the alveoli to collapse. Laplace law, you don't need to know this. This is sort of just extra knowledge, and it really helped me to understand what surface tension was and the purpose of surfactant. It tells us that pressure equals 2 times surface tension divided by radius. And so this shows that for the same surface tension, a smaller alveolus creates a higher pressure than if it had a larger radius. That means that smaller alveoli are more likely to collapse as they require more pressure to remain open or the same shape. So those molecules are closer together, the cohesive forces are really creating a large amount of pressure on smaller alveoli. As well, it's still present in large alveoli, but it's more likely that smaller alveoli are to collapse. Therefore, we have surfactant. Surfactant is a lipoprotein, which means that it has phospholipids and specific proteins within it. So it is an amphipathic molecule. This means that it has hydrophilic heads and hydrophobic tails, the same way you see in phospholipid bilayers of our cell membranes. Now, this reduces surface tension at the alveoli. Essentially, what it does is the hydrophilic heads line up along the surface of the liquid, and the hydrophobic tails are sticking out. So if you look at this image here, you can see this phospholipid layer, that you have these heads on top of the surface of the liquid, and the tails are pointing out because they're hydrophobic. And this equalizes the pressure between large and small alveoli. It reduces pressure more drastically in smaller alveoli, and this is because the surfactant molecules are more densely packed. So there's no pressure gradient between the small and the large alveoli. If there was a pressure gradient, the small alveoli would have high pressure, the large alveoli would have low pressure. And that would mean that when we breathe in, the small alveoli would not fill and the large alveoli would fill, because the air would go from high to low pressure down the pressure gradient. This would mean that parts of our capillary network surrounding those small alveoli would not get the chance to complete gas exchange. And this would be much less efficient. It would cause us to feel fatigued because we're not getting enough oxygen and our body is riding in carbon dioxide. We do not want this. So we equalize this pressure by, okay, so if you imagine a balloon and you were to put dots, so if you have a balloon like this and you were to draw dots across the surface, sorry, this is not a great drawing, but you get the idea. If you were to blow the balloon up, those dots would become further apart from each other. And that's what happens with the with the surfactant molecules, is that in small alveoli, they are more densely packed. This means that when the air initially fills the alveoli, they expand at a much faster rate because there's no surface tension preventing them from expanding. And the large alveoli have more surface tension, so they expand at a slower rate. Once they become equal size, they expand at the same rate, and so that is why we do not have differences in terms of how much the alveoli fill. So the rates of expansion are equalized across all the alveoli. So the smaller units expand faster at the start until they're equal with the larger units. This is a term that you probably do not need to know, but atelectasis is the collapse of alveoli, and especially like the smaller units, because of that pressure gradient. And in this diagram here, you can see the type 2 pneumocyte secreting that surfactant, and the type 1 pneumocyte just being a thin cell. The capillary is here, so this is where the gas exchange would occur across. Okay, we're going to do a types of epithelial tissue recap. Trachea has pseudostratified columna. It is the most ciliated out of all of the gas exchange parts that we've looked at. The bronchus has pseudostratified columna as well. The epithelial cells are also ciliated. The bronchioles are simple columnar and simple cuboidal, so no pseudostratification there. And they are the least ciliated out of the trachea, the bronchus, and the bronchioles. Alveoli not ciliated whatsoever. They have simple squamous epithelium, as well as type 2 pneumocytes, which are simple cuboidal. And now we're moving on to the last part, which is recognizing different sections of the gas exchange system that we've learned about, but through photomicrographs and electron micrographs. Now here we can see a capillary. I know it's from the pancreas, however, I did want to show you this distinct shape of a red blood cell here. So you can see the capillary, the lumen, and inside here, the red blood cell has had to fold in on itself. I know it does have a biconcave shape, but it has folded a little more being within the capillary. Here we see type 2 cells, which secrete the surfactant, and type 1 cells. Now, it's relatively difficult to be able to recognize the difference between the two. You can see a bit of surfactant coming out in this vesicle here, and you can see that the type 1 cells are much more flattened, and their cytoplasm is, you can see it's sort of like this type of whitey, yellowy, creamy color, compared to here the type 2 cells have more purple dots within them. You cannot take this as, don't write in your notes, purple dots in type 2 cells, because the dyes may be different in the images that they give you, and in general, the type 1 cells and type 2 cells are very difficult to distinguish, and you will have a hard time trying to even see the features of the squamous epithelium, which we can see in these other images here, is that we have type 1 pneumocyte here, which is the squamous epithelium. We have the blood cells and the capillaries as well. Here, type 1 pneumocyte again, and here type 1 cell. So whilst this part here is the alveoli, because it has the alveolar space, and here is the lining, you can't really see the distinct shape of that we saw in this image here, with the cell being flattened and the nuclei bulging. So I would say most likely they will not give you questions on this, but if you were to try and recognize, make sure that you first of all identify the alveolar space, and you see that there is one thin layer for the squamous epithelium. Moving on to recognizing goblet cells and ciliated epithelium. These are a little bit more easy to recognize because we can see here the pseudostratified columnar epithelium of the trachea. Here we have the lumen. You can see the cilia are like little hairs sort of on the surface of the cells. And the goblet cell here is quite difficult to recognize when you're looking at the rest of the ciliated epithelial cells. But you can see this vesicle sort of bulging out, and the need for the mucus to exit the goblet cell. Now, if you were to ask why there's difference, if they were to ask you why there's differences in the dyes across an epithelial layer, which is one of the past paper questions that I've come across, you could say that the reason is the goblet cells, as well as different chemicals being used for the dyes, But the main thing is that the goblet cells are filled with mucus, whereas the ciliated cells are not. So that is the main distinction you have to make. Here we can see we have a mucus gland, which I don't believe you should be asked to recognize. It's not on the criteria, but if you were, you can see it's sort of this circular type of image, and you should be able to see there's a slight vesicle in the center of that circle. Now, over here, I believe this is an electron micrograph as it's black and white, but you can see more clearly that the goblet cells are full of these secretory vesicles that are then rising to the surface. And we can see that right in action over here, compared to the regular ciliated epithelial cells, which just have the cilia, and they are not filled with mucus or mucin. Now, this is the last slide. Recognizing alveoli, vessels, and bronchioles. This is not a distinct image showing you exactly how to recognize vessels and bronchioles and alveoli. However, I did want to show you this because I've seen a couple of past paper questions with similar images, and it was really difficult to be able to recognize what is actually going on and identify the different parts. So here, the bronchiole, you can see it has a sort of thicker lining than the rest of the alveoli here. And the blood vessel, whilst you cannot see the distinct 3 layers that you would expect in a vessel, you sort of just have to see that the filling is not air, which you would get in the alveoli and the bronchiole. Because the blood vessel is filled with plasma and blood cells, you can see these sacs are very irregularly shaped. And that's one thing that can help you identify them, that there's a thin layer with these little folds and a very irregular pattern within their outline. So I hope this video helped you understand gas exchange, and I hope the two of them together give you everything that you need to go and start answering past paper questions and link gas exchange with other topics in this year, such as transport in mammals. So if you have any questions, you can comment down below or email me at this is my email, so therevisionvaultsia@gmail.com. And feel free to get in touch if you have any topics that you'd prefer me to cover or any feedback. Thank you for watching, and I hope this was helpful.